computer. Okay, Steve, it's done. Now, thanks to Steve, we have salvaged some recordings that, for whatever reason, they've been lost, and so we essentially have everything recorded up to date. And I have on my YouTube channel the first four. I have the intro to botany, I have the plant taxonomy, classification, I have, or systematics, and then I have the two plant anatomies and morphologies. And I think, well, I haven't put up the others yet, but I have reviewed them all, I think, and Steve did a wonderful salvage job of, because I have yet to explain why they were lost. The audios were there, but the videos weren't, and I don't understand that. But I'm going to be try to stay on top of it, including this one, and find out, because after it's over, it keeps recording, and I look and it's there. But then I look again and it wasn't there. So I'm going to move it from the Zoom file, try to remember to do that, into a file that represents this, a folder that represents these sessions. Okay, so now there's at least one person that's viewing the stuff that's not in his class, because she has a conflict on Sunday morning sometimes. But bottom line is, if you want to review any of these, it's hard enough for me to sit through them. I can only imagine what it's like for you, but to resit through them. But at least you could stop them and start them. That's an advantage in a YouTube. And if you want to do that, just let me know, and I'll invite you by email. And the invite, as we found out, sometimes goes to a social folder in Google. And I think you almost have to have Gmail to make it work, I think. But Steve, correct me if I'm wrong. But anyway, all right, let's get started. And before we get started, I'm going to bring up something that, wait a minute, I've got to share my screen. Oh, boy.
Love your hat, Bobby.
What's that? Oh, my hat. Yeah. If you get it close to read it, it's kind of funny. It's sort of a military sort of thing, but yeah, I laugh about it every time I look in the mirror. But all right, now let's see, what am I, boy. All right, share screen. Well, what's going on? I'm trying to, I don't want to share that. What is, okay. All right, now, oh boy, here my ankles pop. This is something that came up while I was preparing for this. And we're gonna, nowhere in the plant physiology book, you'll find osmosis as a term that's used, and we'll use it today. But nowhere will you find in any plant physiology book I've got the dialysis. Now, most people, when think of dialysis, think of this second meaning. But this first meaning up here, and this is from the Merriam-Webster dictionary online. The separation of substances, dialysis, the separation of substances in solution by means of their unequal diffusion through a semi-permeable membrane. Now, in a living system, it's technically a selectively permeable membrane, because it can determine what comes in and what doesn't come in. A membrane is a living entity, and can actually determine what is allowed in a cell and what's allowed out of a cell. Whereas a semi-permeable membrane might be like some dialysis tubing that you use in an experiment, but it simulates a living system. But anyway, what I like to think of as osmosis is the movement of solute, usually water, through a membrane. Whereas dialysis, I tend to think of it as the movement of the solutes, like the sugar in the water. And down here, they actually go so far as to say, especially colloidal substances, which are larger particles that are suspended. But bottom line is, I might use the term dialysis today, even though technically I've never seen it used in a plant physiology context, but I still think it's appropriate. But now let me go ahead and try this annotate business. Get that going. Where is it? It's been so long I've forgotten how to do this. Where's that little gadget? Nope. Uh-oh. Where's, here it is, Spotlight. All right, now. All right, let me get the PowerPoint up. And actually this is from the last time, and I wanted to make clear about this. This is where we're actually going to start today, but this is where we should have wound up. And you basically see what I call anemic soybeans, what happens when you don't have nitrogen. And related to this is magnesium. Now rhizobium is a bacterium that fixes atmospheric nitrogen in a form that plants can utilize. So the irony is it's 80% of the atmosphere, but the plants can't use it in that form. They can't take it right out of the atmosphere. They have to be fixed in the soil in some way. And then rhizobium is something you find in leguminous plants in the nodules of the roots. Other plants have other ways of doing it. It can be taken up as ammonia and all, but the bottom line is that nitrogen is tied to magnesium. And that's because if you Google tetrapyrrole, P-Y-R-R-O-L-E, you'll get a picture of you'll get a picture of a hemoglobin molecule and you get a picture of a chlorophyll molecule. The center part of a hemoglobin molecule is iron. The center of a similar looking molecule in chlorophyll is magnesium. So, and that tetrapyrrole molecule has a lot of nitrogen in it. All right, here's John. All right, let me just a minute. Hey, John, are you trying to get in? John? Hey, John, you trying to get in? I hung up. All right, maybe he's trying. Let's see. I don't see him. He must be having technical difficulties. I don't know. Maybe I'll try to call him back. But John can never communicate. I have to communicate with him on the phone. But at least he's alive. And even though y'all aren't talking, I'm assuming y'all are alive too. But anyhow, all right. So here we go. Now, this. All right. Acquiring nutrients and resources and moving them through the plants. All right, here's John again. Let's see what happens. Yes, John. Yes. Okay, I'll go back and try. I may have to stop screen sharing to do it. I should be able to mute you, allow you in here, but I'm going to stop the screen sharing and see if I can do it because I don't see you in the. I know you got invited, but I don't see you in the admin realm. I'll try stop the screen sharing and see if I can do it. Hang on. All right, here you are. Let's see. It says family, and that must be you. All right, so now I'm going to start the screen sharing again. Sorry about that. I just didn't recognize the family. You usually don't. Okay, we'll see what. Bye. All right. All right. So now, okay. Well, back to screen sharing. I guess I'm still sharing. Oh, no, I'm not. Here it goes. All right. Now, sorry about that. I hope he's with us. I don't see him here, but I guess if I go down, there he is, family. Okay. All right. Now, all right. So, John, I just told him that it might be as late as the 5th, 13th of June that we do this again because of various factors. But anyway, all right, transport in plants occurs on 3 levels: the uptake and loss of water and solutes by individual cells. And then there's the short distance transport from substance from cell to cell at the level of tissues or organs. And then there's the long distance dispersal or transport of sap within the xylem and phloem at the level of the whole plant. Now, it's not too early to go ahead and say that when we say sap in the xylem, we're talking about water and mineral type sap. When we talk about sap in the phloem, we're talking about sucrose type sap, sugary sap. Now, the next few slides are going to be devoted to this, but before we do that, this other stuff, the uptake, this is about taking, it's not too early to mention that, I guess, but maybe in the next slide, I'll actually get into that. But the next slide, even though this says the following slides is about this, the very next slide could be more about, if you look at these plants, it's more about number one. This has to do with, all right, the question is, why is the plant on the left wilted and the one on the right not wilted? And the answer may not be as simple as the wilted one needs water. Some of you, I guess this is rhetorical because you're on, most of you, Rana's not on, she could actually speak if she wanted to, but that's fine. If you ever heard about burning the roots by adding, usually that happens when people put fertilizer, don't add water. Okay. And so I did it out there in the yard. I didn't burn them, but I made sure I had plenty of water, because if you add fertilizer to the ground without adding sufficient water, what happens? The water will leave by osmosis, leave the plant cells and go out into the soil. And then the plant will wilt. And if you do it, if you're not careful, you'll kill it. By burning up the roots, so to speak. Burnt, end quote, burnt. It's not really burnt, but it'll have a burnt appearance if you look at the roots later. When you go fertilize something, you have to add a heck of a lot of water at the same time, with some loss into the fertilizer, into the stream system, unfortunately, which is what I got against golf courses and cornfields. But anyway, so this is what can happen when you do that. And getting the resources in from the soil, abiotic environment, which is actually, even we call it an abiotic environment, it's full of microbes and it's full of nematodes, which are microbes too. And so it's inorganic matter out there and some organic matter out there. But that's about uptake. And we're going to spend our time talking about mostly 2 and 3. Now, all right, transpiration is in the xylem and translocation is in the phloem. Unfortunately, the internet tries to, when you talk about xylem and phloem, they'll start telling you that xylem is the movement of water and minerals throughout the plant. And that's right. But then they'll tell you that phloem is the movement of sugars and prokaryotic photosynthesis down into the root system. Well, it can be the other direction too. In the spring of the year, the stored nutrients are moving out of the root system up into the upper parts that are becoming non-dormant, becoming active. And so the internet always reminds me, it's like cloud sourcing. If you perpetuate a lie long enough, it gets even worse by the cloud. I think cloud sourcing makes that worse, in my opinion. And Steve knows enough to correct me if I'm wrong. And maybe the rest of you do too. Anyway, but number 2 is about xylem and number 3 is about phloem. Number 3 is a little more complicated because the xylem can do what it does dead, whereas the phloem has to be alive, and we'll talk about that. But a walled cell with a greater solute concentration than the surroundings will be turgid or firm, because if it has a lot of sugars or other solutes in it, water will go into the cell and make it not flaccid, but turgid. And that has a lot to do with the hydrostatic skeleton system in a plant, that if it's limp like the one on the left, you can see it's not turgid, but the one on the right is turgid. Now, so part of the structure of the skeletal system in a plant is, besides xylem, is the very fact that they call it a hydrostatic skeleton. Water in turgid cells makes it not wilted, makes it look normal. So in other words, healthy plants are turgid most of the time. As turgor contributes to support in the non-woody parts of the plant. And that's important because the non-woody parts don't have hard walls yet. And that's why the herbaceous appearance depends on water to add to that hydrostatic skeleton. All right, so the interactions of water and the solutes, that's the dissolved substances in a solution, including minerals, are the subject of the next few slides. Anatomically involves the xylem and the phloem. Okay. Now, transpiration is, as it says here, the evaporation from the leaf surface by way of the stomata. Stomata is the plural of stoma. Stoma is really means mouth. There is the rest of the fusion evaporation. Okay. Technically, transpiration is through the substomatal chambers below the surface of the leaf, but some loss of water takes place directly through the epidermis and by evaporation. And that has a lot to do with how thick the cuticle is. There's a waxy substance. You know what? I need to go back and get my annotate button. Shoot. All right. If there's a real waxy cuticle, some plants have waxier ones than others. That cuts down on water loss. Hairs cut down on water loss too. The tricky part of a plant, and we'll talk about this later, is that most of the water it needs to operate to get that 10%, to get that amount of water, it loses a lot. So now, okay, the adhesion, cohesion, tension, transpirational pull theory is a way to explain how this comes about. There's for a long time, and it's quite involved, and we'll go into it, but there is other processes called root pressure that really doesn't get water up very far. In fact, I was looking this morning and didn't see it, but I have a hydrangea. Sometimes you'll see marginal water droplets around the leaf, and that's known as guttation, guttation. And guttation is a function of root pressure. That, however, is not enough to get water to the top of a 150, 200, or even 300-foot tree. Because you just can't generate that kind of root pressure to push water that high. But you can create a little vacuum, which is what this kind of does, and get it and accomplish it. Transpiration actually gets water up to the top, but also cools the plant and maintains turgor pressure. That's what that hydrostatic skeleton was talking about. And the rate of transpiration depends on light, temperature, and air movement, like wind. And I'll show you a device called a potometer in a moment that can measure it. And it's regulated by the guard cells that surround the stoma. Okay, any questions? I'm having to do that. Now let me try it. There's a trick to making that start working in Zoom, I've learned. Let's see. Okay, I used to ask this question on a plant physiology test, and you don't have to worry about it. And I would tell the students beforehand that I was going to ask it, and they still wouldn't get it right because they just, some of them wouldn't. But describe the path of a water molecule from the soil as it moves through the plant and moves up out here and enters the atmosphere. And that takes about a paragraph to do it. And I think I even did it for some reason last time, or ahead of myself. But when you've got, I'm going to use it to explain what's going on here. But when water, actually, next slide, let me move ahead let's to here, because all right, let's say a water molecule, and I'll come back. A water molecule is out here in the soil, and it comes in through the epidermis, through a root hair, and into the cortex, which is what this is. And then when it starts to get, if it comes in next to the phloem, it's going to get blocked by the endodermal tissue here with suberin in it, a waterproofing. You don't want to put water in the phloem. You want to put water in the xylem, because that's what's going to conduct it up. So by osmosis and dialysis type activities, well, water is going to move by osmosis, and the minerals, if they're in solution, will move through too. But basically, they're going to move into the arm, and this has 4 arms. It's not always 4. This shows 3, but this is a buttercup root cross section. It's been stained, and these thick wall cells are xylem conducting elements, or actually in some cases like tracheids. But anyway, and then once the water gets in there and the minerals get in it, they'll start moving up. And that's once you get to the pericycle, which is best defined by the layer inside of the endodermis, that's right here, which gives rise to lateral roots, by the way. That's another function of the pericycle, the main function. Once it gets in the vascular skeleton of the root, it moves through the transition zone into the stem and on up into the leaves. Now, back to this. All right, we've got water moving up through this column, and the adhesion, cohesion, actually, looks like I shortchanged it here, the cohesion tension. Technically, it's also adhesion, cohesion, tension. I never realized I screwed that up, but it looks like I have. Technically, it should be the adhesion, cohesion. Maybe they just call it that with the understanding adhesion is a part of it. Okay, here's the deal. First, cohesion is the sticking together of water molecules, and hydrogen bonds do it, although individually they're weak, collectively they're strong. And so you have water molecules, it's kind of like, did anybody ever watch Woody Allen, Everything You Want to Know About Sex, We're Afraid to Ask, or something like that? But anyway, they had these sperm cells lined up on a plane about to jump out. And bottom line is that the water molecules are going to jump out into the atmosphere, but they're all connected to one another in this long chain. And when one comes out, another comes behind it. So, but part of it, that's cohesion, attraction of like molecules to one another. But the adhesion has to do with the water molecules adhering to the colloidal or cellulose walls. So not only are they stuck to one another, they're stuck to the side of this wall, and they move up in this column. Of course, if you stuck a knife in here and embedded it in here deep, you would have put air into the system and ruined the whole thing. So it's kind of like a little vacuum effect. The xylem tubes form continuous strings extending from up into the leaves all the way down into the roots. And before a water molecule can actually leave the leaf for the substomatal chamber, which is this little area right here, and going from a liquid to a vapor state, which it does inside the leaf, it doesn't do it. I mean, by the time it builds up some vapor in here, excuse me, let me drink some coffee. There's a vapor pressure deficit that gets created between the inside here and the outside, and the vapor water vapor will move from where it's more concentrated to where it's less concentrated out here. Creates another vapor pressure deficit in here, and the water molecules continue to move out. Now, this happens, has a lot to do with these guard cells here. One showing the nuclei, which, let's see, I have a better look at them. Well, actually, it's pretty neat to see the veins. These are veins, xylems, with the thickenings in the walls. But let's see if it's the next slide shows it. No. I guess it's several. I'm looking, I know I got a graphic somewhere of guard cells. I guess, I don't remember where it is, but it's sure not here. But two little sausage-like cells, if you could look at them, look like little curved sausages if you look at them from the side. So the whole thing is called the stomatal apparatus, and part of the stomatal apparatus is two guard cells. For this to work right, the guard cells have to be open. That means, in other words, to get water through, and actually to get carbon dioxide in, and to let the oxygen out from photosynthesis. Now, to make it work right, there has to be accumulation of one of the ingredients in the NPK of fertilizer, the K being potassium. Now, the sad thing, and for every advantage there's a disadvantage, and some leaf, MOLDS, leaf molds secrete a chemical that causes the guard cells to accumulate the potassium ions and thus stay open so that the fungus can enter the cell and infect the plant. What a dirty deed, something without a brain that can do that. But that's a little aside that bears out that advantage to disadvantage. For every advantage is a disadvantage. Now we use this already, and this was about trying to show you that the endodermis is thick, is opposite the phloem, and that's what that red staining stuff is about here. It's not much here, and it's not much here, because you want to direct water into the xylem, not put water in drown the phloem. You're not trying to do that. That's what the Casparian strips are called. These are the Casparian strips. Actually, I could move this away a little bit. Well, I'll try to. I guess I can't. Well, I can't do it with that annotate thing there. But anyhow, maybe I could minimize that. Oh, okay. Water solutes move into the vascular cylinder by passing through the cytoplasm of endodermal cells. That's what's going on there. Now, this little fancy piece of glass is a device called a potometer. I don't like not being able to see that y'all are there. So let's see if I can toggle back. Well, I lost everybody. I lost myself even. I don't know how to get myself back. Geez, I'm sorry about my ineptitude here. Wait a minute. There we go. I don't like that angle. Now I'm able to control it. I couldn't control it a minute ago. Anyhow, all right, the potometer. I can't tell you how many times I've broken one of these things and how expensive they are. I don't even know what they cost now, but they're not cheap. And what they can do in the lab is, if you take your plant that you're using, herbaceous plant, one that's not too woody, cut the sucker off diagonally underwater, and diagonally increases the surface area. You do the cutting of this underwater, and then you make the fitting with what I used, paraffin or something. I forgot what I used. It's Vaseline, I believe, to help make the seal tight, airtight. You don't want to get air bubbles in here. And then you have a reservoir of water, and what you can do is, if this will be graduated like in milliliters or cc's, same thing, milliliter, cc, cubic centimeter. And you can actually measure over time the movement of water. And you can turn a fan on and speed it up, or you can turn the fan off. You can make the room humid and slow it down. So you can actually measure the rate of water loss this way. And you can demonstrate it most qualitatively and measure it quantitatively. And the sad thing is 98, well, some say 90 to 98% of the water that enters the plant is lost through the chute system by evaporation. But the evaporative loss includes transpiration, and some of it straight through the epidermis itself. All right. Now I'm having to mess this. Okay, here we go. All right. Now translocation. Again, this number 2 can xylem can function. The sapwood can function in conduction of water and minerals in a dead state. Of course, the heartwood is no longer functional in conduction because the cells are occluded, so it does function in support. But unlike the xylem, the phloem has to be living to do its job. So in other words, phloem cells are functionally alive, which means they can move sugars and other dissolved substances, and they can do it both ways. And the companion cell acts like a bridge tender on a bridge. It can direct what moves through the sieve cells, or in the case of angiosperm, sieve tube elements. Now, I made this statement in passing back when we did anatomy, but I'm going to repeat it here. Compensation point was that point physiologically when photosynthesis and respiration are equal. The plant burns everything it makes, but and we talked about that concept, but back then, actually, when we started plant physiology, when I was talking about the biochemistry dealing with photosynthesis and respiration. But the truth is, the transport of sugar from the leaf, not photosynthesis, limits plant yields. In other words, if you don't get rid of the sugar from where it accumulates, you're not gonna, that's gonna slow the whole process down. And the plant yields are the net increases in biomass. Carbohydrates is what we're talking. The phloem transports the organic products through the plant by a process called translocation. We already said that, but the pressure flow model describes how it happens. It's also considered a hypothesis, the model. And then to discuss it, we need to entertain the concepts of a sugar source and a sugar sink with the details on the next couple of slides. Now, in angiosperms, the specialized cells of the phloem that do it are called sieve tube members. Sieve tube members are the counterparts to the vessel elements. Sieve tubes are counterparts to the vessels. Sieve tube members are cells, they're microscopic. Vessel elements are cells, they're microscopic, but you can actually see vessels with your naked eye in wood if you know what you're looking at. And if you really know what you're looking at, you can see sieve tube members to some degree with your naked eye. Sieve tubes and vessel elements in the xylem run inches or even feet sometimes. And in terms of linear up and down in the plant. But gymnosperms do not have vessel elements. Actually, there's one group that does, highly evolved gymnosperms. Now, but in general, gymnosperms don't have vessel elements and vessels. And gymnosperms do not have sieve tube members. They have something called sieve cells. And sieve cells in the phloem and tracheids in the xylem are not as efficient as the conducting their counterparts in the angiosperms. Not only are sieve tube members and vessel elements larger in diameter, it's like a garden hose compared to a conduit, a culvert out there. I mean, think of a garden hose versus a 2 or 3 feet in diameter type concept. So sieve tube elements are hollow, except they've got these shower drain-like entities between, let's see. Here we go. All right, see the sieve tube members here, sieve tube member here, and sieve plates. Now xylem doesn't have over here that there's no shower drain between them, and these plates can get filled up and become non-functional. And the companion cell directs the activity of the movement. So let's go back. Okay, so I'm actually trying to tell you that one of the reasons gymnosperms aren't as successful today as the more highly evolved angiosperms has to do with their lack of the sophisticated conducting tissues in the xylem and the phloem. All right. Okay, let's see. C graph. Okay, next slide. We're doing that now. Whoops. Shoot, this thing is real sensitive. Okay, now. Let's see. All right. Let's see if there's anything different here. Water contains phloem. Phloem sap is aqueous solution, which sucrose disaccharide is most prevalent. Solute, and to understand solute, you have to think about a solution. Solution is made up of solvent, usually water, and a dissolved substance like sugar, the solute. So a solution has those two components. The phloem sap may also contain minerals, amino acids, and hormones in addition. All right, but this is one of the better graphics comparing the xylem, water conducting cells of xylem, as opposed to the sugar conducting cells of the phloem. This movement is, as I already said, is up. This movement here can be both ways, even at the same time. It can be up and down. But seasonally, in this early spring, it was going to be mostly from the roots up. And as the sugar gets made in the leaves and stored down below, it would be coming down. But it could happen. It has to do with the sink and source and sink concept that we're going to deal with now. And this first bullet is what I've said at least twice already. All right, now the sugar source is a plant organ, especially mature leaves, in which sugar is being produced by either photosynthesis or the breakdown of starch. Okay, now we think about it. We said C6H12O6 glucose is made by the process of photosynthesis. But the excesses get stored as starch. To make starch, you have to do a process we'll mention in a minute called dehydration synthesis. But the opposite is breaking down starch by the reverse process known as hydrolysis, splitting by water of a big carbohydrate. And a sink is simply an organ such as a growing root or tip or shoot or tip or a fruit that is a net consumer or storer of the sucrose, which is the sugar. Now the next slide gets into. I'm going to let you read these bullets, or maybe I'll read them to you because, well, you can read them. Well, I'm going to go. I'll go over these now, then I'll actually go over some bullets. The upper leaves on a branch may send sugar to the growing tip, whereas the lower leaves of the same branch export sugar to the roots. One sieve tube may carry sap in one direction, while the adjoining a different tube may, in the same vascular vein, may flow in the opposite direction. The direction in each depends on the locations of the source and sink of that tube. Now, a sugar sink usually receives its sugar from the sources nearest to it. Okay, a sugar sink like this receives its sugar from the sources nearest to it. Phloem cells at the source actually transport by the ATP energy expenditure sugar into the sieve tube members. Okay, they put sugar in sieve tube members. This increase in sugars causes a solute, SOLUTE potential to become more negative, which forces water to move into the sieve tubes and creates a pressure that pushes the sugar and the water down, whatever direction is needed. As a result, water this causes an osmotic effect. Okay, when the sugar goes in, the water is going to follow, just like the fertilizer scenario I was describing. And when the sugar and the water move in, the pressure buildup is what creates a source. So the combination of sugar and water molecules cause a pressure increase in the sieve tubes and pushes the material, mostly sucrose, through the tube, through these sieve plates. Pressure flow. Down here at the sink end, plant cells remove sugars from the sieve tubes out into the joining cells, and takes energy to do that. And then the water follows, and so that creates, and the water moves too. So that creates a lowering of pressure at the sink end. In short, the building of water pressure, hydrostatic pressure at the source end, and the lowering at that pressure at the sink end allows water to move from a source to the sink down to, in that direction. That's about as complicated a thing as we'll ever talk about, but that's what's called the sink, the pressure flow model or hypothesis I just tried to describe. And even though I'm not a plant physiologist, I didn't do too bad, I guess. I may hear myself on tape and think otherwise. Okay, now, a storage organ like a tuber, that's an Irish potato, or a bulb, an onion, may either be a source or sink depending on the season. When the storage organ is stockpiling carbohydrates during the summer, it's a sugar sink. Now, there is something that's interesting to me. Well, actually, it can be talked about here. Carbohydrates like glucose eventually get stored in animals, it's glycogen, which is animal starch, but in plants, it's carbohydrates, more complex carbohydrates like the regular starch. And the precursor of starch is sucrose. All right, glucose is a volatile source of carbon that you either burn it in respiration or you store it as, or you move it, you combine two monosaccharides like glucose and fructose and make sucrose, which is the way it gets moved through the plant. So 3 levels of carbohydrates here. You have the simple monosaccharides like glucose and fructose, which are isomers of one another. Put a glucose and fructose together and you make sucrose, which is the primary way that carbohydrates move through plants. But they're the most stable way to store them is to convert them by dehydration synthesis into a starch molecule. Now, if you've got a little piece of paper, write this down on a piece of paper, C6H12O6. C6H12O6, that could represent glucose or fructose, or actually even a galactose molecule. They're all isomers, but they're structurally different. So if you put a glucose and a fructose together, you make C12H22O11. That's sucrose, C12H22O11. If you look at it carefully, well, gee, how come it's not C12H24O12? That's what double C6H12O6 would be. But what I'm trying to tell you is sucrose is deficient in H2O. And that's what dehydration synthesis is all about. To combine, here's the bottom line, people, to combine a glucose molecule with a fructose molecule to make sucrose, you have to remove a molecule of water. So sucrose has H2O removed to make it. So you keep doing that over and over again, and you make not a disaccharide like sucrose, but a polysaccharide like starch. I think that's, pretty neat when you get down to it. But sorry for that digression, but it was actually, it helps you to understand what dehydration synthesis is all about. And again, other solutes like minerals are also transported in the sinks, and amino acids too, along with the sugar. All right. Now, the concept of totipotency we first introduced when back in plant anatomy. Before we go though, a lot of that stuff was pretty heavy. I hope the fact that there's no questions doesn't mean you're lost. But
tell me back on the sieve tube members, what did the gymnosperms have instead? I missed that.
Okay, good. That's good. All right, Donna's question has to do with angiosperms versus gymnosperms and the differences in the anatomy. All right, what you see here is in angiosperms, both in the xylem and the phloem. In the gymnosperms, instead of having vessel elements, which are open end to end, all the gymnosperms have in the xylem is tracheids. That means that the water has to, watch my cursor, has to go like this. Zigzag, very inefficiently go up. But in an angiosperm is high diddle diddle right up the middle. Not only is a vessel bigger in diameter than a tracheid, it is open-ended. In other words, a vessel element is a more differentiated xylem parenchyma cell that has really differentiated in hollow openings and in wider diameter. Whereas a tracheid is differentiated a little bit, but has bordered pits that water moves zigzag its way up. It's a very circuitous route for water. Now for the phloem, in gymnosperms, there are no sieve, there's only sieve cells. There's no such thing as sieve tube members that are joined to end to form sieve tubes. And so the movement of water in the xylem and sugars in the phloem is less efficient in the gymnosperms. Did I answer your question, Donna?
Yeah, I think so.
Yeah, you're probably not the only one that had it, but anyway, Donna's like me. She doesn't, she doesn't worry about it. The older you get, the less you care about what people think, so you're liable to ask any questions. So that's good. Not Donna, I'm not trying to date you as being old, but anyway, that's the nice thing about you don't really. You asked a 104-year-old lady what she liked about being 104. She said no peer pressure. So, anyway, now the concept of totipotency. I'm glad Donna brought that up because that to me is a lot about evolution. So, has a lot to do with why one of the reasons why angiosperms are more dominant today than the gymnosperms. And the age of the mammals is the age of the angiosperms, and the age of the dinosaurs is the age of the gymnosperms, including cycads, which I like cycads. Okay, the concept of totipotency. We first introduced this term back in the plant anatomy when we're running slow. I'm running, I better, anyhow, the parenchyma cells are totipotent, and the pericycle cells in the root are totipotent. And we mainly find the parenchyma cells in the pith, and we find them in the cortex, but they're like the stem cells of animals. Now, so we're going to study growth and development, but we really should say, according to some plant physiologists, we're going to measure growth and we're going to watch development. That's the way they like to say it. But now, the Botany for Dummies book that I talked about a long time ago on page 163 says something like this. At the cellular level, plant growth results from two different processes: cell division by the meristems and growth by the addition of new cells. That cell division is addition of new cells. Another aspect of growth is the elongation of the cells as the cell expands. Okay. Now, this is an experiment showing what happens when you take phloem cells, companion cells that have nuclei in them. Sieve tubes lose their nuclei at maturity. Sieve tube elements do. When you take phloem cells from a carrot and put them in a tissue medium, the proper medium, and of course aseptic technique, so you don't want any moles or other parasites to get in here. And then you put it in the right medium and you can regenerate an entirely new carrot. And that's a manifestation of the totipotency of these parenchyma cells taken from the phloem. And this again is, if we could get animals to act like, to do the stem cell thing better, we could regenerate. Well, actually, we're starting to have some successful regeneration of limbs and things, but now, of course, a drawback to this process, any cloning process, is everything is like everything else. There's this is what happens. That's what happened in 1846 in Ireland when they propagated potatoes by cloning the buds of the eyes. The whole island of Ireland was had a genetically identical population of potatoes that was susceptible to the late bite of potato fungus Phytophthora infestans. So once you do cloning, you make everything, you don't get the genetic variability, you get sexual reproduction, and you put all your eggs in one genetic basket, and you could potentially have a problem. My favorite little saying, Hadaway-ism, is asexual reproduction is a short-term asset or short-term gain, but it's a long-term liability. So when you propagate things asexually, which I suppose any sex is better than no sex, but if you do it enough, you eventually cut down on genetic variability, whether it's artificially doing it like this or whether it's in some plants can do it. Like got African violets in there that are over 50 years old. And the reason that they're doing that is they've asexually reproduced through the years. Now, this process is called in vitro because it's in glassware. The counterpart is in vivo in living systems. But this is a callus. A callus is a mass of undifferentiated cells, basically parenchyma cells, totipotent cells, and put in an aseptic situation where this is free of disease. And it looks just like a mass of cells here, and that's what it is. Over time, this callus differentiates into a new plant. Now, this is a neat experiment because next time we'll introduce plant hormones, and I'll go ahead and say right now that there's two categories of them, auxins and cytokinins. And this experiment has, like every good experiment, has a control, and it has four variations of the experiment. Now, this is looking at what it takes to get the right ratio of auxins to cytokinins, or cytokinins to auxins. When you have the control here, you get a callus like here in the left that stays. And by the way, this is no accident. The pith is a good source of parenchyma cells to do it. And so this callus, which is an undifferentiated mass of parenchyma cells, totipotent cells, when you subject that in that test tube, you should put a cytokinin ratio to auxin that is high. You get the stem growth and no root growth. When you do a low cytokinin to auxin ratio, you get the roots, but nothing up here, no stem leaf growth. And then down here, when you use an intermediate ratio of cytokinin to a low auxin concentration, you get the callus continues to stay a callus. It grows without differentiating. However, the successful in this, when you want to get this kind of result, where you get this kind of activity and this kind of activity in the same callus, you use an intermediate ratio of cytokinin to auxin. And this is the combination that produces a new plant. Okay. Okay, so it has to be the right ratio, right combination to get successful cloning. Imagine how much experimentation that took first time. Now we're going to look at seed anatomy and germination, and before we do germination, we'll try to review anatomy. In some cases, not a review, I guess, but we're showing 3 seed types here. If you look up what this is, a caryopsis, Caryopsis in Harris, it'll tell you that a caryopsis is a one-seeded indehiscent fruit that has the fruit wall joined to the seed coats. And they'll have grain as an example. And the grass family is what they're talking about. So a corn kernel or kernel and grain are the same thing. A kernel of corn is actually a one-seeded indehiscent fruit that has the seed wall fused to the fruit wall. And in corn, we're going to see several representations of this in the next few slides, not the very next one, but we're going to talk about something called, it'll be written on the next slide. The cotyledon is called a scutellum, Scutellum. It's an absorptive cotyledon. The cotyledon is a seed leaf. And then we have the apical, the stem region here and the root region here. The coleoptile is a sheath over the apical meristem. There's something down here called a coleorhiza over the root, kind of like a root cap but this whole thing is an embryo. This whole entity in a dicot is an embryo. There's the root. And this is a baby here, a baby plant. And then this is something the sad thing about doing this Zoom session, you really can't have a real lab. And because in lab, I would take the lima beans and soak them overnight. And of course you can see it in a peanut, but not as well. But if you soak a lima bean overnight, it really imbibes, takes on water, and you can see the little baby. But this is castor bean. It's not a true bean. A true bean is in the legume family. Phaseolus is a genus. And it's not too early to point out that the food source, endosperm, all right, endosperm, we're jumping ahead of ourselves because most of you don't know what endosperm really is. And toward the end of the course, when we talk reproduction, which is we're getting there, we'll say that in a pollen tube, there are 2 sperm. And there are 2 sperm in gymnosperms and 2 sperm in angiosperms. So one of those sperms fertilizes the egg. The other sperm fertilizes something that we'll talk about called a polar nuclei, the 2 polar nuclei, which it actually was on an earlier slide in an earlier presentation, but I put it off. We haven't really dealt with it yet. But the bottom line, another reason that makes angiosperms more surpassing the gymnosperms today is they have a food source that the gymnosperms don't have. This one sperm does the sex thing with the egg, but the other sperm fertilizes the polar nuclei and makes a food source that's very nutritious. It's called endosperm. In the case of most angiosperms, or at least dicots, like you see here, the endosperm is not stored in seed leaves called cotyledons. These cotyledons with the little veins actually look kind of like leaves, real leaves. Whereas over here in a true bean, the endosperm has been stored in the food source is not really in the form of endosperm, but it's stored in the cotyledon. This so-called seed leaf is actually very fleshy. So there's 2 primary differences in the way dicots store their food source. So the castor bean, the bulk of the surrounding embryo the material surrounding the embryo is endosperm, and this endosperm will be absorbed by this root system, and then eventually the leaves will kind of wither. We'll see. Now, seed anatomy, there's three primary components here. The embryo, which consists of the cotyledons, the epicotyl, and the hypocotyl, and the radicle. Epi and hypocotyl are regions. The region above the point of attachment of the cotyledons is called the epicotyl. The region below the point of attachment of the cotyledons is called the hypocotyl. The tip of the hypocotyl is the primary root or radicule. Now over here, this one doesn't have the endosperm, and this one does. And the endosperm is actually in these cotyledons, kind of like what's left and right, similar to what's left and right here. Now, let's see. All right, so that's one component of the seed. The second component is, well, technically it's the endosperm on this one is separate from the embryo. Over here, it's a part of the embryo. And then the final component is a seed coat. Now you say, what seed coat? What are you talking about? Well, think of Spanish peanut and the little red skin that's on them. That is the seed coat. An ovule becomes a seed after fertilization. The covering on an ovule is called integuments. After the integuments mature after fertilization, the integuments are labeled seed coats, and the red skin on the Spanish peanut is a good example. Now, this is from University of Georgia. I took it from them and give them credit here. They're showing fir, which is a pine relative, and they're polycotyledonous. They're many cotyledons, and that's typical. And let's go through the drill here. Seeds develop and to allow reproduction, and this is sexual reproduction. The functions of the seed parts. Endosperm provides energy. Embryo is the baby, the new plant. The seed leaves are a storage form of food. And the hypocotyl is a region that's below the attachment of the cotyledons. The radicle is the tip of it. This is not, the seed coats are protection. And then down here, that's not shown here, but in this fact, gymnosperms don't show a very good epicotyl and plumule. That's the young apical meristem right there, the baby. In fact, this is one of my favorite slides because it shows, we're coming up on an hour, but we're actually getting closer to the end quicker than I thought. If on a lima bean, you go and soak that sucker up and take off that seed coat, it comes right off when you get it wet. You can actually, with a hand lens, or sometimes with your naked eye, see an opening called the micropyle. Does anybody know what the micropyle does and is? Okay, well, the micropyle is the place where the sperm actually enters to fertilize the egg inside. The hilum is a scar that's left by the funiculus, which is the equivalent of an umbilical cord that held a bean to the pod, and it's pretty interesting. Then the rafes region or scar that's up here, and calliza is a ridge. But anyway, so this is the anatomy of a bean is a lot more than meets the eye. And this is one of the downsides of not having a lab. Notice that in this up here part, by the way, the plumule is the region, the tip, the young, where the bud is, and the epicotyl is the part of attachment of the embryo above the cotyledons. Notice that how big the cotyledon is and how small the embryo is. Down here, once it starts in the ground, the cotyledons are small, and the seedling, which was an embryo, is quite large. So notice the size differences between these cotyledons and that. All right, now this and this are the same. It's just that it's colored and flipped over. But the question we could ask you of this is this a photograph of a monocot or dicot? And with the two cotyledons, it's obviously what? A dicot, whereas a monocot has one cotyledon. Now, this happens to be shepherd's purse in the mustard family, and the epicotyle actually shows the young plumule. It's not labeled, that's the bud, and you can see that the root develops much quicker than the stem, and that's because it has to anchor and draw up water and minerals for this thing to do anything when it gets in the ground. A suspensor and a basal cell is something that can be demonstrated in some cases. It has to do with absorption. Now, corn, I said I'd show you a more detailed look at corn. This is a kernel or caryopsis. You can't see it very well here. You can see it a little bit better here after this is flipped over. And when this is a sectional stain, so this, what you see here is the same thing you see in the center. The orientation's the same. If we flip this over 180 degrees, it would look like that. You can see the scutellum is the absorptive type of cotyledon, and a corn kernel is absorbing all this nutrients in a sperm and bring it into this embryo. This is the embryo with its scutellum. This is the coleoptile is a sheath over the apical meristem. The coleorhiza is a sheath over the root, kind of analogous to the root cap, but it's something monocots have. This is even better view in the diagram. The graphics are even better. It just shows more detail, and the labeling is good. So pericarp is a 50-cent word for the fruit wall. Pericarp, think of it this way: on an orange, the rind is the exocarp part of there's three parts to a pericarp. Peri means around, carp means fruit. It's not easy to think about in a corn kernel, but in an orange or banana, it's easier. But an orange is good because the rind is the exocarp. The fleshy part you eat is called the mesocarp, and the endocarp is that pulpy stuff in the center. So sometimes in like a peach, there's some fusion of those carp parts to each other. Now, we're running up on an hour and 5 minutes. I think I'll be finished in the next 8 or 10. Okay, there are external and internal factors that contribute to germination. Germination is when a seed becomes non-dormant, becomes active. And depending on the plant, they can be years, sometimes in a few cases, 1,000 years, but that's unusual. There's this saying about annuals and perennial seeds. Woody plants tend to have short-lived seeds where some annuals have long-lived seeds. The conditions have to be right, external and internal, for that to happen. And the usual process, if you've got to get water into that seed, and in a lab, what we do, we take some honey locust seeds and file them because they're so tough that they have to be filed to get water in them sometimes. And you say, in nature, how's that happen? Well, it's not often, but sometimes it's a matter of running down a stream and having the seed coat abraded to the point that water can come in. You can put a light Clorox solution and eat away too to break away some of that seed coat. Now, sometimes passing through the gut of an animal will have enough acid to affect the seed coat, and that's by the evolutionary design. The coevolution going on there. To get a seed to germinate, sometimes they have to pass through the gut of an animal to make it happen. Okay, let's see. Amount of stewed stored may be insufficient, and the seed may abort. Dormancy period, not right length of time. Those are all factors, and global warming can affect some of this too. we believe. Oxygen, okay, if you don't get, you can't burn any sugar without oxygen. Well, you can, but it's called anaerobic, and you don't get much ATP energy out of it. So it's a very inefficient process. But to get seeds to germinate, generally you got to have a sufficient supply of oxygen. So it's a combination interaction of external and internal factors to get seed germination. Now, seed takes up enough water. That's called imbibition, IMBIBITION, imbibing water. And that's, we would demonstrate that in the lab, and the students would actually file away the seed coat on the seed to get some, the next day or next period, we actually see it. Okay, now, okay, the seed germinating, and here about to get this plumule and this epicotyl and plumule growth, that there's quite a bit of going on down here already that you don't see. This is just a graphic comparing monocots and dicots, and in corn, the scutellum remains within the seed. In the case of bean, it emerges above ground. In the pea, we'll see in a moment, the cotyledons stay below the ground. This is called epigeal, but this is called hypogeal germination. And that's what's going on here. The corn has the cotyledons and the seed stays down below. In the case of the pea, it also stays down below. But unlike its bean, the cotyledons are up here and they're withering. So this is called, both of these have hypogeal germination. Hypo means below, and this has epigeal germination. It means the cotyledons come up above. So, and the cotyledons are withering away at this point. It's interesting that these leaves are opposite on this young seedling, but in pea, they are, or bean and pea, in the legume family in general, the leaves are alternate. Same is true of this beech seedling with the cotyledons here, and over on this castor bean, the cotyledons are withering. But these are really come out, the primary leaves are opposite, which is the basic condition. But the phyllotaxy is really alternate on beech. And the bad joke associated with this is students who are out there in the beech forest, what are these baby things called? And they'd say, beech seedlings. No, and I'd say they're sons of beeches. So, all right, now, actually we're done. And so it didn't go too badly. Let's pull the plug on it right here. And we got one more plant physiology to suffer through, I hope. And that'll be, as I said, probably is the likely time will be the 15th, 13th of June. Let me unshare this screen. Forgotten how to do this. Jeez. All right, let me shut that off. All right. Well, that's what happens when you don't do this often, you forget. I can't unshare. Steve, help me. Meeting controls. Well, jeez. Oh, gosh, sorry about that. Just a few weeks and I've forgotten. But anyway, that's a little embarrassing, but like I said, I shouldn't care. I'm old. All right. You can unmute and ask any questions or have any comments. And so I will join Rana and see if, okay. All right.
Question?
Any questions?
Yeah, building on Donna's question about the
Yes.
sieve tubes and lack thereof, angiosperms to
Yeah.
gymnosperms. Is that related to why when you cut a pine tree, it oozes real thick, sticky sap versus when you cut, say, a tulip poplar, it'll ooze water?
Well, they actually are losing more than water in the tulip poplar. I don't think there's a connection between what you just described. In fact, you can cut a vine. You might see water dripping even from a grapevine. You can actually, you know how in the jungle they'll cut a vine and drink it. Well, actually, I can go out there and do that with a grapevine. But you're getting water visibly, but you're actually getting an adjacent, the inner bark, the phloem. You're getting sugars, but you're seeing the obvious thing you're seeing is the water. Now, back to the pine. Pine, if you do a cross section of the stem, you're going to see that they have something that gymnosperms don't have a lot of, and it's called resin canals. If you cross section a piece of pine wood, you'll see the typical thing we'd see. You see cortical tissue and all that, but eventually it starts hardening. But even when it hardens, there are canals that are just, you can see them with your naked eye practically. They're loaded with resin. So it's not, that's what Donna's asked doesn't have a lot to do with what you're seeing, I don't think, Steve.
Okay, thank you.
That's a good question, though. I mean, I'm not trying to minimize it. If we had a course in plant anatomy, we would study you'd see more about what I'm talking about. And the anatomy of a pine needle, I talked about it a little bit, it's really designed to be a drought tolerant plant because the veins in the heart of a needle are in the center, away from the surface, whereas the substomatal chambers of angiosperm leaves are near the surface and more subject to in general, gymnosperms are more drought tolerant than angiosperms because of that. Okay, all right, everybody. So we had everybody but Jackie and her husband. If you ever get a chance to do the between March and now in the desert in Arizona, you should, and that's what they're doing. All right, I'll seriously consider the next time trying to do the polyclave key. A couple of you have it already, and if you want, I'll send it to you. But it's not necessary that you go through it while I go through it. You're seeing me do it and you doing it is two different things. But I'll rather than have everybody go pick the same plant in the yard, which is impossible, all the way from Naples, Florida to the mountains of North Georgia or the Piedmont. Speaking of that, by the way, if you don't have the Guide to the Vascular Plants of the Blue Ridge it's an old book by Eugene Wofford, one of the guy I went to school with. This is, if you look at the map, it covers Cobb County and it covers Cherokee County. And that's about as far down as it goes. It goes all the way up to Virginia. But the keys in this book, there's no pictures. The keys in this book, even though the thing is outdated in a way, are excellent. They're short and sweet, and they just work like a charm. And I went to school with that boy, and he's now not a boy anyway, he's retired. He was a curator of the herbarium at University of Tennessee, or was. But I spent many days in the field with him as a young person. And anyway, but that made me think of that when I said Piedmont. Okay, all right. So bye-bye, everybody.
Bye-bye, and thank you, Bobby.
You're welcome.
Bye.
Bye-bye.